Hello viewers, we all know the basic structure of the double helix as proposed by Watson and Crick. Now, depending upon certain parameters of this basic DNA molecule, it can take different conformations, and the three helical conformations, they are the A, B and Z DNA conformation, about which we are trying to understand why these conformational changes takes place and what is the significance to the biological world of these various helical conformations. That is, what are they and why they are in different conformations. So the three families of the DNA helix, they are the A, B and Z DNA, and majority of the DNA is in the form of B DNA, that we know is the double helical Watson-Crick model. Now of the three, the A and B, they are the right-handed helix. When we say right-handed helix, we mean that if we face the helix towards our face, it is clockwise. The direction of the helix, if it is clockwise, it is the right-handed helix, and if it is anti-clockwise, as is the case in Z DNA, it is referred to as the left-handed helix. These various families of DNA helix, they have particular biological role, they are of significance, and they are essential, they are interchangeable, all these three forms, and they help in the various basic functioning of the DNA molecule, like the replication, the gene expression and the recombination phenomena. They also play different roles in the formation of the DNA chromosomal structure, that is the nucleosomes and the supercoiling of DNA and all. Now we say that these three forms, they are interconvertible. So there are certain conditions that determine which form of the helix will be present. There are three basic conditions. First is the ionic or hydration environment. The second is the superhelical tension of the DNA molecule, and this tension is because of the specific DNA sequences. So we may say that these three forms depend upon the particular DNA sequences that are present in the stretch of the DNA. And the third thing is presence of specific proteins which bind to the DNA in one helical conformation, and that results in the change in the conformation. When we say the ionic or hydration environment, the A DNA is favored by low hydration, while the Z DNA is favored by high salt concentration in the environment. Similarly, in case of DNA sequences, so the A DNA sequence is favored by certain stretches of purines, or in certain cases even pyrimidines. Stretches of purines like this GA, triple GA, guanine, adenine. So stretches of guanine, they induces the Z-DNA conformation. Similarly, these alternating purine-pyrimidine nitrogen bases in the stretch of DNA, they induce the formation of Z-DNA. Now, if we talk of the basic structure of the DNA, we know that there is the phospho-di-ester backbone, and from it the nitrogen bases they stick out. This phospho-di-ester backbone is basically negatively charged. This is a characteristic feature of the DNA molecule, and all the characteristic features that we are going to talk about in this video, they play a vital role in determining the conformation of the DNA molecule. So these phosphate groups present in the backbone, they are responsible for the negative charge of this DNA molecule. And in order to neutralize this negative charge of the DNA molecule, certain ions they are made use of, like the sodium ions. Then various polyamines like spermine, spermidine, they also neutralize the negative charge. Various divalent cations like magnesium ions, and of course the DNA binding proteins, the positively charged, the basic proteins, that is the histones, which are rich in amino acids like lysine and arginine. They neutralize the negative charge of the phosphodiester backbone of DNA molecule. Now, second part of a DNA molecule is the nitrogen base. Nitrogen bases that are attached to the sugar moiety at the C1 prime carbon of the sugar this way. So the overall orientation of these molecules, of the sugar molecule, of the nitrogen base molecule, the organic molecules in space within the molecule, that is of basic importance in determining the conformation of DNA molecule on the whole. So here we are going to talk about two molecules. The sugar molecule having the 5 carbons, and if we consider this plane of sugar molecule, that is the plane defined by this C1 prime, the oxygen, and the C4 prime. In reference to this plane, if the C2 prime lies above this plane, then the conformation is C2 prime endo conformation. And if this C3 prime carbon, it lies above the plane that is formed by this C1 prime, oxygen, and C4 prime, then the conformation is said to be the C3 prime endo conformation. That means orientation of this 2 prime carbon or 3 prime carbon in relation to this plane of C1 prime, oxygen, and C4 prime. Now, one thing is clear, that if this C3 prime part is above this plane, then the distance between the C3 prime and 5 prime carbon is reduced, and this will be the case in the C3' endoconformation. So second thing that is very much obvious from these two figures is the orientation of the guanine, the position of the guanine molecule. This guanine molecule can be positioned in two forms. One is the antiposition and the other is the syn position. So in the syn position, the guanine molecule is nearer to the phosphate entity, to the backbone entity of this, and this happens in the Z-DNA, as we are going to see in the video. Now, since this C2' endoconformation is in B form and the C3' conformation endoconformation is in A form, and we have seen that in case of C3' conformation, the distance between the phosphate group and the 3' carbon reduces. This flattens up the molecule. It makes the helix broader in case of A form. And second thing is that these nitrogen bases, they are shifted towards the phospho-di-ester backbone, and this leads to the formation of a hole within this DNA helix when it is in A conformation, and the diameter of this hole is approximately 9 angstroms. Now one more thing is there regarding the nitrogen bases, and that is the propeller twist. Propeller twist means the angle with which the two nitrogen nucleotides, nitrogen bases which form the hydrogen bonds, which are connected by means of these hydrogen bonds, they are not in the same plane. They are twisted a bit, and that is referred to as the propeller twist. While twist, when we use the word twist, we mean the angle between two adjacent nitrogen base pairs. So that is the twist, and it is because why this twist take place. This reason for this twist is that these nitrogen bases, they are hydrophobic, and since they are hydrophobic, the twist, they are not stacked one above the other. They are stacked with a certain angle that induces more hydrophobicity in this molecule of DNA, in the internal part of this DNA molecule. The third term that we have to understand over here is the pitch of the helix. That means in the helical structure, the length of a full turn of a helix, that is 360 degree. So what is the length of the DNA molecule, and that depends on the sequences that are present because of this twist, which is determined by the adjoining nitrogen base pairs. Now the basic reason for the helical structure, why this DNA molecule is a helix, So, as we have seen in the previous slide, it is the hydrophobic nature of these nitrogen bases because of which the twist is there to maximize these hydrophobic interactions, and that leads when two adjoining bases they are going to have a twist. Gradually the entire molecule in which the stacking of these nitrogen base pairs will take the form of a helix. So that is the reason for the helical structure of the DNA molecule. Now, second thing that we talk of when we uh, describe a DNA molecule is the presence of major and the minor grooves. Major and minor grooves refer to the distance between these sugar molecules. Now, in a if we talk of these two parts of the DNA molecule, the two opposite strands, this is the backbone and here is the nitrogen base pair. So, a part of the nitrogen base pair is more near the sugar, and the sugar entities, the distance between these sugar entities is less. So the side on which the distance between these sugar entities is less is the minor groove, whereas the side where this distance between the sugar entities is more is the major groove. And for a particular base pair, the position of the major groove is always the same, because it depends on the structure of these pyrimidine and purines. In case of A and T, this is the major groove, adenine and thymine, and this side will be the minor groove. Now, the three conformations, the A, B and Z, they differ in the dimensions of these major and minor grooves, and also the role. In B-DNA helix, we know they are clearly defined major and minor grooves. So, specific sequence, the proteins which bind to specific sequence of nucleotides of the base pairs, they are present in the major groove, while the minor groove they provide the site of attachment for the proteins which bind to the DNA molecule non-specifically. Furthermore, these minor grooves they are the site of attachment of various water molecules and other small ions also. The size or the dimension of the major groove of bDNA is enough that it may adjust a complete pyrimidine, the single cycle nitrogen base, not the dicyclic purines which are. So it is deep enough to form a triplex DNA molecule if some nucleotides fit over here. In case of a DNA molecule, the size of the two grooves is more or less the same. It is a stretched DNA. While in case of Z-DNA, the minor groove it is very deep and narrow, while one cannot easily distinguish the presence of a major groove. It is almost non-existent. Now this Z-DNA, it is a very typical sort of conformation of the DNA. Although it is a high energy form of DNA double helix, but it is stabilized by negative supercoiling. It is left-handed DNA molecule. Now, the helix repeats of the Z-DNA is approximately 12 base pair per turn. That means for one 360-degree helix, left-handed helix, 12 base pairs have to be present. And the average rise distance between these two adjoining base pairs is 3.7 angstrom. Now, in contrast to this bDNA, where the nitrogen base pair, they are the repeating unit. In case of Z-DNA, two base pairs, a dimer is a repeating unit. That means per helix repeat, there are only 6 dimers, 6 basic units per turn present in case of Z-DNA molecule. And what is the reason why the size of the repeating unit is 2 base pairs in Z-DNA? That is because of the conformation of the sugar and the glycosidic bonds. In case of Z-DNA, the C2' endoconformation is in anti this cytosine or thymine deoxyribonucleotides, while in case of G and A, guanine and adenine, the conformation of the sugar molecule is C3'. In case of A and B, it was either C3' endoconformation or C2' endoconformation. While in case of Z-DNA, whether it will be C2' endoconformation or C3' endoconformation will depend upon the type and orientation of the nitrogen base present. So that gives the zigzag formation. The twist angle of the, we know that Z-DNA is in the part of the stretch of nucleotides where they are repeating CG base pairs. So the twist angle is 9° between cytosine phosphate guanine, whereas if the order is reversed, that is, we always talk of from 5' to 3' region. So if it is guanine phosphate cytosine step, then it is 51°. So when we say that there is a repeating unit of 2 base pairs, so the total twist angle of a repeating unit is 60° in case of Z-DNA. Now this zigzag path that is adopted by the DNA molecule in case of Z-DNA, it is because of the electrostatic interactions. Since the backbone is a zigzag path, the phosphate groups are closer to each other and they are negatively charged. So because of the electrostatic repulsion between them is more. So if we want to have a stable Z-DNA, we need more of high salt, more of ions. So we say that Z-DNA is stabilized in high salt concentration because it needs more of positive ions to stabilize the electrostatic repulsion between closely lying phosphate groups. Now, these are the various parameters which differentiate the three forms of DNA. The helical turns, that is, whether it is left-handed or right-handed. Then what will be the diameter, depending upon the your C2 prime endoconformation and the C3 prime endoconformation. Then the height of the helical turn, that is the pitch of the helix, and how many number of base pairs are present in the helical turn. Then the helical twist per base pair, that will ultimately lead to the helical structure of the molecule. Then the distance between each base pair, that is helical rise per base pair. The base tilt to the normal helix. Then the dimensions of the major groove and the minor groove. The carbon pucker, that is the ribose sugar conformation, which is C3 prime endo in case of A DNA, C2 prime in case of B DNA, while in case of Z DNA, that depends on whether it is pyrimidine or purine. And the glycosidic bond conformation, that means whether they are the anti-conformation of the nitrogen bases or the anti for pyrimidine and syn for purine, as is the case in Z-DNA. Now, if we quickly go through the significance of the A-DNA and Z-DNA, this A-DNA, it protects the DNA molecule from UV radiations, as is seen in the sporulating bacteria, because the nucleobases in A-DNA, they are less susceptible to UV damage. Then it forms this A-DNA, it forms various complexes with the polymerase, the DNA polymerase. Its structure, it improves the way these bases, the nitrogen bases, they fit with the template primer duplex, and this way it helps in the formation of the various complexes with RNA polymerase as well as DNA polymerase. Then in case of E. coli, we know there are cap binding sites where this cyclic AMP receptor proteins they bind, and that is also facilitated by the A form of DNA, which shortens the distance because it is more broader between various sites of this cap binding protein. Then the data box binding proteins, that is also facilitated by the DNA conformation of DNA molecule. Now, it is seen that in the long terminal repeats of transposable elements also, these stretches of DNA, they are of common occurrence. These DNA stretches are present wherever there is a hybrid of DNA and RNA molecule in the transcripts that are being formed in the uh, post-transcriptional modification part, or where there is double-stranded RNAs. At that point also, A form of helix is observed. Now, a transition from this eDNA to aDNA, it also occurs during the transcription, because again at the point of transcription a DNA RNA hybrid is formed. So that will give a more stable complex formation if the form of DNA is A form. Now, initially it was considered that this Z-DNA is not present in the cells. It is basically observed only under high salt conditions, under lab conditions, that is in vitro. But in vivo, they have been observed and they have got their own biological significance. So, they are more abundant in eukaryotes, because in eukaryotes, in the promoter region, where this cytosine phosphate guanine islands, they are present in clusters. Over there, these DNA configuration, that is the Z-DNA configuration, they are formed. So they are present in the promoters of various genes. They help in the transcription. They are present in the form of transcription enhancers, because these Z-DNAs, they they assist in the activity of various topoisomerase enzymes. So they are present in the form of transcriptional enhancers and in terminal differentiation points also. They also help in RNA editing of the proteins. They also bind to the Z-DNA stretches of the DNA molecule. So in this way, the various conformational forms of DNA are because of the variations in the basic parameters of the DNA molecule, and each form has got its own biological significance. Thank you.